Dear Sir and Madam, Organizing Committee and participants on conference, I am going to represent you my report, Software for Simulation of Bio-like Systems and Processes Using Movable Cellular Automata. I am Katarina Gazduk, the PhD student of Software for Computer System Department, Yuri Fedkovich, Chernivtsi National University, and my co-authors are Volodymyr Zhikharevich and Sergei Ostapov. Simulation of biological processes and systems is one of the most relevant and promising areas of scientific research. The mechanisms of many bioprocesses are currently unknown. Therefore, one of the tasks that is solved by simulation is finding answers to questions that are still open. For example, the search for ways of cell organization and the evolution of living matter. Nowadays, simulation problems for biosystems are widely developed and require more complex calculations, so traditional approaches become less effective. Many biological phenomena such as intracellular localization of molecules, morphogenesis, growth, and dynamics of force distribution are related to a spatial component that requires an understanding of spatial temporal systems. Investigation of the localization and distribution of systems components leads to the need to reproduce the shapes and geometry of the studied object as well as their possible deformation over time. In image systems, natural forms, spatial distribution, and their temporal dynamics are derived from images. This interprets the image not only as a visualization but also as a quantitative measurement and makes it a major source of data. However, the use of image as a quantitative dimension raises several key problems. First, we need to investigate and take into account all prior knowledge and hypotheses about the system we are studying. Secondly, we need universal methods that can be applied to more than one specific situation. Finally, we need user-friendly and efficient software that facilitates the transfer to new methods to everyday scientific practice. A review of existing sources had identified a number of software systems that allow simulating of bio-like processes and structures. One of these programs is CompuCell 3D, the creation of which was started in 2004 and improvements to the system are still ongoing. This development allows us to simulate morphogenesis processes in different organisms as well as to visualize the phenomena of cell growth, change in the direction of distribution of chemical, etc. CompuCell 3D is a three-dimensional modeling environment for complex biological processes that integrates several mathematical models. Also, CompuCell 3D provides the ability to test and experiment with biological models by providing a flexible package with many different levels of control. The user must have a broad range of bioinformatics knowledge to use this system. The disadvantage can be attributed to the poor visualization of the simulated system. Other interesting computer simulation system is Morpheus. It is a simulation environment for studying high-scaled systems. Morpheus developed for the study of cellular models under the influence of both internal and extracellular dynamics, which is described in a special declarative language. Using the means of numerical solution of ordinary differential equation, partial differential equation, and cellular model of pods as in the previous CompuCell 3D system. However, despite the wide range of settings and possible functionality, the construction and study of the dynamics of a multicellular organism is not foreseen. Ashalabd is the next CSS that was considered. It's a program for molecular dynamics simulating under a single graphical environment are represented as author's own implementation of molecular dynamics and also a method of classical and quantum mechanics of popular programs. The disadvantages of this software are next. Its application to reflect the dynamics of biosimilar processes is quite limited, since the main area of modeling and related relationships are focused on a chemical, physical process inside a molecular. All models mentioned above have both advantages and disadvantages. Also, it should be noted that there are other software complexes for simulating of bio-like processes that achieve more precise characteristics but are narrowly focused. In contrast to the simulation system mentioned above, we propose development of a universal computer system that would allow researchers to visualize the model not only through a set of built-in interaction functions, but also to create their own. The main purpose of the system is to provide a wide range of users, scientists, engineers, or just interested people a convenient tool for simulating biosimilar processes and structures. This tool should be simple and flexible when used. In this regard, the proposed CAD system is based on the method of movable cellular automata. The core of the system implements an iterative algorithm for selecting automata, searching for the nearest neighbors and their interaction with each other. And the interface part provides visualization of the model and setting of input parameters. Next, we provide a description of the structure and features of the functioning of the CAD system BLSD, bio-like structure design. The system has a modular architecture and is created by means of structured programming. The central part of the system is a procedure that provides the implementation of the algorithm of the method of movable cellular automata. The method involves the operation of an iterative loop. In parallel, there is a graphical representation of a set of movable cellular automata. These two cycles are implemented as separate threads. That is, the system in this respect has a multi-threaded architecture which can significantly increase system performance in terms of multiprocessor hardware. Data structures are implemented in the form of arrays, which store all the necessary information about the models and their parameters in the graphical window. These arrays are read or written to files with extension BLSD. All information about the set of movable cellular automata is stored in a two-dimensional array E, the structure of which is shown in Table 1. The line numbers of the array correspond to the numbers of movable cellular automata in the model. The second column corresponds to the type of automaton. Then, the three next columns contain the x, y, and z coordinates of the automaton in space. The following Nmax columns contain indices of adjacent automata. Finally, the last columns of Pmax contain the possible values of continuous parameter of cellular automata. For example, the sign of the active or passive state of the automaton, the value of some internal energy, the component of the momentum vector, etc. In addition to the described data structure, the system uses structures to store information about the properties of the types of automata as well as the function of their interactions. The names of automata types, their parameters, the names of elementary and complex functions, as well as other text information are also contained in the corresponding arrays of cells of the string type. The system has a multiply window interface. The main window depicted in the left figure is intended for manipulation of the model and consists of two parts: the field of control of the model's operation of the program and the field of output of three-dimensional graphic image of the model. The main menu contains options for invoking standard file, save, or open dialogues, editing rules for interaction rules, and movable automata properties, and also a system information info dialog. In general, the system can operate in two modes: construction and simulation, which is determined by the state of the respective controls of the graphic user interface system. The upper left part of the main window contains buttons for controlling the simulation processes. The properties of movable automata and the rules for their interaction can be edited in the dialogs that appear when you select the appropriate menu item Edit. Consider the window of editing properties of automata shown in the figure right. This window allows one to specify the full number of types of cellular automata that can be used when the model is being implemented. This number is entered in the number of times field and determines the dimension of the matrices of functions shown next. Selecting a specific type of automaton for editing its properties is made, then its number is entered in the field type index. Next, consider editing database of function. This window allows you to form a complete set of complex interaction functions, the indices of which can be entered into the corresponding cells of the interaction matrices. This allows you to define the full range of rules of automated interactions. Complex interaction functions are set or composition of elementary rules. For a complete list of all elementary interaction rules, see the right in the set of elementary function windows. These elementary rules implement the corresponding elementary iterative step to modify the state of the automaton. Next, consider editing the matrices of functions shown in right figure. This window allows you to define the rule of interaction between all possible combination of types of two interacting automata. As mentioned above, the dimensions of the matrices correspond to the number of automaton types in the simulated system. Moreover, since there may be different rules of behavior between unbound and connected automata of the same type, two matrices are also required: matrix for sliding neighbors and matrix for fixed neighbors. It should be noted that example which demonstrate the possibilities of the systems and proposed method were described in our previous investigations. In this paper, we consider the problem of the construction of a CAD system for a simulation biolike structures and processes. A universal computer system for simulation biosimilar processes and structures was developed. The method of movable cellular automata was chosen for the main calculating model of the system. The library of elementary functions, the parameters of which can be customized according to the requirements of the model, were described. As a result, computer models mimicking certain natural phenomena can be obtained using BLSD system. The evolution of the proposed system is expected in two main areas: development of a base of elementary rules and means of their editing, the ability to download a library of elementary rules from the appropriate DLL, which will allow one to develop a database of rules in any programming language, and also enhancing interactive graphics and improving quality. Boost orthographic projection and perspective projection, a variety of graphic effects such as transparency, fog, blur, and more. Also the possibility of group selection of elements and their copying. Thank you for your attention.